Hi, my name is Fazila, and my project is about mitigating metastasis and utilizing cancer-associated fibroblasts to stop the spread of cancer. To give a background about cancer metastasis, cancer is the most common disease diagnosed worldwide. When cancer spreads from one organ to another, it becomes significantly harder to treat, and the health outcome for the patient decreases. This process is known as metastasis and is the primary cause of mortality in most cancers. The metastatic process involves a series of events from proliferation, where the cancer multiplies non-stoply. It ultimately involves internalization of cancer cells into the circulation system, where they travel with the CTCs, also known as the circulating tumor cells, to sites of metastasis. After excavating out of the blood vessels, proliferation causes a second tumor to form in a different location. This series of metastasis cascade can be seen in the image to the right, where the cells proliferate and they invade into the blood vessel and extravate out of the cells in a new site to proliferate and form a secondary tumor site. Any failure to complete one of the single cells in the metastatic cascade would lead to arrest of an entire process. This therefore raises hope for finding drugs to prevent the metastatic cascade by targeting isolated and specific features of metastatic cells. The introduction to my project is the proposal by Stephen Paget, a pathologist in 1989, who proposed a seed and soil theory which emphasizes the concept of tumor spread. His theory is basically is a seed is when a seed is planted, it is carried in all directions, but they can only live and grow if they fall in a congenial soil. Similarly, during a metastasis, malignant cells spread from one tumor site to through the blood vessels or the lymph system to other sites of the body, where the malignant cell form a new tumor and override normal tissue functions. In this case, the seed is the malignant cells or the cancer cells. The congenial soil is the tumor microenvironment. The congenial soil has all the nutrients and that requires for the seeds to grow. And similarly, tumor microenvironments has the necessary proteins, macromolecules for the cancer cells to metastasize. To describe the image to the right, the cancer cells leave the primary tumor site, which is A, and they uh, travel through the circulation system, which is B, along with the CTCs. The circulating tumor cells then see distant sites, which is C and D, and seed metastatic tumors itself, which is E, and then they return back to the primary tumor, which is F. The tight interactions between the tumor microenvironment, or the TME, and the cancer cells form a protective V shape that forms a tumor formation. The TME is mainly composed of multicellular cells from mesenchymal, endothelial, immune cells, pericytes, and fibroblasts, arranged in the extracellular matrix, or the ECM. All these multicellular cells can be seen in the image to the right. Because metastasis presents a serious barrier to effective treatment and recovery, it is crucial to understand how much cancer-associated fibroblasts play a role in the breast cancer metastasis and progression. My project is mainly focusing on CAPS and its interaction between other proteins in the HCM and how it affects the growth of tumor. CAPs are basically activated fibroblasts and is a major component in the tumor stroma. It secretes signaling molecules, also known as cytokines, which supports the tumor growth, invasion, migration, and metastasis. They're often found in the circulation system with the CTCs and help them bind to the endothelial layer. The idea behind the circulation caps and CTCs cluster is that the cancer cell takes the soil with them, which has the necessary proteins and the macromolecules to make it easy to seed a new metastatic site by supporting tumor growth. The image here shows the CAF's internalization and their clustering with the CTCs and metastatic seeding in new tumor sites from the primary tumor. My main project is to target the CAF clusters to prevent metastasis. In the breast cancer microenvironment, non-cancer cells like CAFs cross-talk with the cancer cells to induce many of these events. I discovered that cancers, CAFs, circulate in the patient's blood and clone clusters with CTCs. Using vitro models, the research focuses on signaling crosstalk mechanism underlying CAFs, CTCs internalization, facilitating clusters, and driving role of clusters in metastatic seeding. The purpose of the study was to investigate the physical interactions between CAFs, CTC, and endothelial cells, an important first step in the process of extravasation, wherein a cancer leaves the circulation to initiate a new metastatic site. My hypothesis is that if I target the specific proteins in the ECM that helps the functions of CAFs and CSCs clusters in the blood circulation, then there is a chance to reduce the bindings of CAFs to the endothelial cells and eventually prevent metastasis. While this work focuses on the relationship between CAFs and cancer metastasis, other proteins in the ECM impact the CAFs' productivity on a wide variety of cancer functions, which means that the scope of CAFs' impact on cancer is much larger than that of just metastasis. This project was mostly done remotely at home due to the restrictions of COVID-19 pandemic. To replicate the model of CAF BC cluster extravasation, the endothelial binding assay was done. The endothelial lung was made with a layer of Hewitt cells. The cultured cells was created at height lawn wherein other cells cannot penetrate. The calves and the BC cells were labeled with cell tracker dyes and pipetted on top of the endothelial layer and incubated for 30 to 60 minutes. After washing off, pictures were taken using computer software so that we can count the calves binded to the endothelial layer. My main part of the project was to consisted of differentiating the endothelial and the calf PC cells and counting the fluorescent cells, which is shown in the image to the right. I used the image toolbar, which is shown on below, to and from the Fiji app to keep track of the cells. In each experiment, where different treatments of culture cells were used, the cells were treated with different specific proteins and media. The results from my project was that from different treatments was shown in graph and analyzed. The haze treatment to the left causes the hyaluronic acid degrades, which partially inhibits cap cells binding to the endothelial layer cells. This shows that when the amount of HA reduces in the tumor microenvironment, then the CAF's binding also reduces in the body. The mark cells of the cat without the haze treatment, and they are much more higher than the cat cells with haze treatment. To the right, the oscillatory stress graph presents the flow of blood in the blood vessels. The shear stress affects the calf binding to the endothelial cells, which can be represented in the graph where it shows the comparison of cancer cells and calves placed into rotating platform at 100 RPM at 0 minutes and after 5 minutes. Thus, in our body, if the blood flow increases highly, then the calf binding to the endothelial layer also decreases, which is shown in the graph here. The graph below, which is the 24-hour pre-treatment and the calf media during binding assay, shows the average percent of MDA231, which is an aggressive cancer cell, breast cancer cells, and MCF7, which is a less aggressive breast cancer cells, bound to the endothelial cells in the presence of regular media or calf conditioned media. Both type of cancer cells shows insignificant changes. Thus, in the body, it does not have any effect on the calf binding. In conclusion, the finding reported in this study supported the hypothesis that CAFs and CTCs cluster in the circulation promote the extravasation of CTCs through the physical interaction with endothelial cells. Additionally, the project further showed that the ECM proteins, specifically hyaluronan, is an important for this interaction. More work needs to be done to study how CAF cells influence the expression of other proteins like collagen. It is also important to understand how calves vary from one cancer line to another, for example, a breast cancer to a prostate cancer. This research provides a new perspective on the intersection between calves, the ECM proteins, and the cancer metastasis. 90% of cancer deaths are associated with cancer metastasis, and thus it provides a significant obstacle to effective treatment. So, it is crucial to understand how factors from the tumor microenvironment influences the way cancer spreads. Thank you. These are my references I used in my project and also for the images.